Hello friends, so welcome to the part 1 of lecture 1 for week 9. So now we have discussed the assembly and various intricacies of assembly part very well. Now the next part is doing the functional annotation, right? So functional annotation could be a general purpose thing, like you have one gene and you are doing the annotation, or it could be like you have the entire genome with all new set of genes and you are doing the annotation, right? It could be the genomic set or metagenomic set, right? Sometimes it could be tricky that you are not finding any annotation or functional information about the sequence by conventional methods like BLAST that we'll discuss today, right? And you use alternate methods that we will see in the next part of this lecture. Okay, so why basically we should do the functional annotation of protein? Because proteins do all the major functional tasks in an organism, and they also determine the complexity of the organism, right? So basically they are important to understand the functional characteristics of an organism, and for a new genome it is particularly important because there would be some set of proteins that are basically orthologous or also present in other species, right? There would be some proteins which are species specific, some which are unique to that genome, right? And some for which you will not find any annotation, right? So which basically is limited by the current knowledge, and maybe those are not proteins or doing some unknown function that we don't know. Okay, so basically the process of functional annotation has evolved a lot in the past few decades, and today generally like we should be able to annotate like very large fraction of the set of proteins from any organism or metagenome or any environment. So basically it provides clues about the complexity, also like function and evolution, because proteins are the entities that actually evolve and lead to all the differences in the phenotype that we see. So evolution in proteins can tell a lot about the genomic clues about an observed phenotype. Okay, and also for drug designs, drug target, treatment of disease, because many a times we are looking for proteins which could be drug targets in microbes, right, which can selectively kill a microbe or a virus, okay, but without affecting the human host or other hosts, right? So drug design and drug target, these are like very important also functions for this. And then when we know like what proteins are important to us, we can do engineering or basically designing of those protein modifications and designing, so that in silico, so that we can improvise their function in a very directional manner, right? And also like their cell to cell interaction and like in pathways they are very much involved, so that understanding is important. So basically for a function we should know that where does a protein bind, what reaction it is catalyzing, which pathway it is coming from or belongs to, whether it is common between pathways or unique to that pathway, like as an enzyme. Okay, like in the cell, what cellular function it is doing, okay, what other proteins it is interacting with, because protein interaction is also very important area to understand a disease or problem, right? And where it is exactly found in the cell, its nuclear location or membrane or basically cytoplasmic location, and the structure of it, which is also very from like several decades we are trying to generate as many structures as possible for proteins, so that if we know this structure, we know the active sites, we know its regulation, allosteric or other things, right? So that way we can understand more about the proteins. So one of the key methods to determine the function of a protein is by BLAST, or basic local alignment search tool. Okay, so basically it is a local alignment search tool, so it does not try to align like this is your protein sequence, this is like some sequence that is matching to it. It will not try to align completely, it will try to align in parts, let a local similarity. If the two are identical, of course it will lead to like to ultimate result will be like end-to-end similarity you will find. But sometimes or basically I would say that most of the times for non-identical proteins it would be only some pieces that will match more, and that is captured by the local alignment. It can be done for nucleotide or the protein sequences, and there are like dedicated databases, non-redundant protein and nuclear databases that can do this job. Okay, and it can tell the like functional evolutionary relationship between proteins. It can do the taxonomic assignment. Okay, so basically if you see here, two types of alignment are possible. One is global, where these are sequence 1 and sequence 2, and we are trying to align from head to tail and try to see, like, even if there are some regions which are basically not matching, right, but the ones which are matching we are trying to align, right. So we are trying to stretch the alignment from head to tail. Okay, whatever is aligning we are aligning, and whatever is not aligning we are keeping as gaps or unmatched regions, right. Okay, so that will basically ultimately will translate into some kind of score. But in local we do not do this. We will align only the region which is the best matching one between two proteins or two nucleotides, right. So the advantage and disadvantage of both the approaches are there, but generally, like in local alignment, it can be quick, right, and if you are able to align a considerable part of a sequence, right, with a good identity and or similarity and like score and e-value, like these things, right, it is more likely that the two proteins will be doing the similar function. Okay, so basically pairwise, and actually both global and local alignment are pairwise. Even if in global, basically we are trying to align from head to tail, and we can do this for multiple sequences also. It is done initially by the pairwise alignment. Okay, and local alignment is always like pairwise. Okay, so basically local alignment, we are trying to look for regions of maximum similarity within these subsequences, right. Okay, so like in global we are trying to align the two sequences, and even if there are gaps in between, okay, and this is a match region, we will continue aligning, whereas in local we will only align the parts which are actually aligning, and the remaining parts we will skip. Okay, so basically this slide tells like broadly like what is the advantage, disadvantage. So in local alignment we are trying to identify short conserved sequences, and in global we are trying to do the entire alignment. So the local will be good for identifying like small conserved or basically more similar identical sequences between two sequences, and that is why it is good for functional annotation and taxonomic assignment. Function, because it is matching to a protein in a database, so whatever function that protein is doing, your protein which showed the match should also do the same function. And similarly, whatever is the basically taxonomic annotation of that protein, like whichever is the closest matching protein you have found or sequence you have found in the database, so whatever genome or host basically organism it is belonging to, you can try to see that basically your taxonomic assignment of your protein should also be similar to that. It could be at the same species level or at the genus level or family level, LCA based approach that we will see later. Okay, so basically local cannot identify short like each and every residue that is conserved, but regions, whereas global can identify even single residues which are conserved at a like with gaps on both sides. Okay, so that is why it can be more useful to identify conserved residues which could be more important for the criticalities of the protein function. So here complete alignment is not done and also not the objective, but here we try to align from head to tail, and that is why it may take more time, the global alignment. Okay, and basically in local of course we will miss out on some conserved residues, whereas in global we can cover those. And for local the algorithm is Smith-Waterman, whereas in the global it is a dynamic programming algorithm which is Needleman-Wunsch algorithm. Okay, so these things we will see later. Okay, so BLAST is one of the local alignment size tools which is designed for speed, accuracy, as well as reporting results which can be validated later also. And also it can determine the long distance relationship if you do psi BLAST and other kinds of BLAST. Okay, and that is why basically it is very useful for functional annotation. So it was developed by Archul et al. In 1990, and since then it has evolved a lot. There are some other methods of annotation also, which are not as sensitive and specific like BLAST, but they can do the job well. Like they can be more like much 100 or like 1000 times faster sometime, and can be used for like mass level of annotation. Okay, but BLAST is like the gold standard for the annotation. So we'll see like how the BLAST algorithm works. So basically you have a protein which is your query and a protein which is in the database, right? Okay, so basically what it is doing is it is trying to break the protein into seeds or small sequences. Okay, so it does not try to align like from head to tail, but it breaks your query into small seeds. Okay, for example, word size could be 3 to 4. Okay, 3 to 4 amino acids, for example, right? And then basically it will try to align those seeds with the database protein. Okay, so basically and it is basically what happens is, it is not that you will find the exact 3 or 4 mer that you have derived from here, okay, in the database. There could always be some chances that some residues may be more conserved, some nucleotides or proteins, amino acids should be more conserved, some are less. Okay, so you may not find the exact match. So that is why you create a library or list of words which are similar to the original word. Okay, list of words, right? Okay, so basically what it is doing, it is creating a library of similar words to each of the seed, and then trying to match that with the database protein. And of course, while it is making the library, there could be several combinations possible, because you can rearrange each and every position with all the 20 amino acids, but we will only retain those list of words which are above a particular threshold. So when we go further, we'll understand this more. So once it has found a match, okay, it will try to check that match in both the directions. Okay, like if this is found, it will try to extend this match in both the directions, right? So this is what I was saying. So basically this is your query sequence, right? Okay, so basically we will break this query into overlapping fashion, such that DRV, then RVA, then VAT, and then ATS and TSL, and like that we will create a list of words. Okay, now for each of these words, okay, that we have broken, right, we will make a list of similar words, like replacing each position by all the 20 amino acids. So this way we will have a list of words for each of the word. Okay, like DRV can be DRI or DIV or IRV or like that. Okay, so all possible 20 amino acids can be replaced at the 3 positions in that seed. Okay, and then in the database protein, it will try to match that seed. So basically, for example, here it was DRV in the query protein, whereas in database it is VRV. So VRV can be matched with DRV, but it is not the exact match, but a partial or basically similar match. So that will be given a score. Okay, if you get the best match, you will get the highest possible score. If it is a partial match, then you will get the lower score. Okay, that we will see in the next slides. And once you have matched it, you will try to match the adjacent residues also, as in 3 mers or 4 mers or whatever is your seed size, also using the same logic of partially similar words, right? Okay, and then try to extend in both directions and keep on extending till you are finding a match in the sequence, right, database sequence. Okay, so now the list of words that I have created, for example, RGD can be KGD, QGD, RGE, by changing one amino acid at each of the position, right, from all the 20. So that will get a score, because if the initial word is RGD, okay, so RGD matches with RGD, gets the highest score, R with R, G with D, and D with D. Okay, and if R is changed by K, then the score will be 14, for example. Okay, because R to K match gets some score, G to G and D to D will get some score. Okay, so there are 3 positions. Okay, and each position will have a score of match. If it is a complete match, you will get the score. Okay, so now what is the score thing? So there is tables like this is PAM 200 and Blossom 62 matrices, scoring matrices. Okay, so where there is a score is written like if I replace K by R in a protein, okay, then what is the match mismatch score? Okay, if I replace K with D in that protein, then what is the resultant score? But if K is retained as K, then what is the resultant score? Okay, and this score is actually derived by the alignment of all the known proteins and from different protein families, and looking at that what all changes are more acceptable in the nature or in the sequences, and what can be rarely be changed, like glycine if you change by something like phenylalanine or tryptophan. Okay, that is not a good change. But if polar with polar, non-polar with non-polar, hydrophobic, not hydrophobic amino acid, so those things will get a more better score. Okay, so this way, like for each position you will create a score for that list of word. Okay, like RGD with RGD got 17. Okay, so RGD, if I do RGD, so basically all three positions will get some score. Okay, so basically total I have to make 16, so it could be like 8 or 5, 13 and like 4, 17. Okay, similarly if I change it by KGD, okay, so it could be like 5 plus 4, and basically you have to make 14, so it will be what, 5. Okay, so this is just random example I am giving. So basically if art is changed with K, it is like 5. Just random example, it is not from the table scoring matrix. Okay, so this way we will have these, and we will retain only those words which are above a particular threshold. For example, okay, so that threshold is inbuilt. Okay, so only not all the combinations of 3 words will be tried, but only like if the top score is 18 PQG with PQG, right? So up to 13 whatever list of words is possible will be considered for the possible match with the database protein. Okay, so PQG matched here, right? It was PMG here and PQG here, and PMG had a score of 13. So this position will get a score of 13. Okay, and then the next MGS and QGQ will be matched, and similarly this side KTP, TPQ and TPM will be matched, and like same partial match criteria. If they show the good match, basically it will be keep on extending in both directions like this. Okay, and this is called the high scoring pair. Okay, and high scoring pair is nothing but basically you are extending the match to both directions, and keep on extending till you are not able to find further matches, or there are significant like gaps in the two sequences, or unaligned regions in two sequences. Okay, so again going through this, so list of words PQG, QGE, GEF, and like, okay, in a overlapping fashion by shifting by position by 1. Okay, and for each of this we'll create a library of word PQG, a library of words, and that library of word we are matching in the database sequence, right? Like PEG is matching here, and the query was PQG. Okay, and giving the score for that. Okay, so basically if you go stepwise, so the first step is when you get a sequence, remove all the low complexity regions or repetitive regions, because they will complicate the process of alignment. Okay, because they can be found anywhere in the sequence randomly, so they should be removed first. So we will remove the low complexity regions, then make a K letter word list. Okay, and for each word you will make a list of all the possible matching words and arrange them into, like, based on score, into an efficient search tree. Okay, and basically you match the database for the exact matches of the topmost or the above the threshold list of high scoring words. Okay, and then when you find a match, you try to see the neighboring positions also in that sequence if they are also matching, and this way it will continue and your match will be extended till it can be extended, okay, till you find the match in the database sequence. Okay, and this way you make a high scoring pair, and basically it could be that one seed was matched here, one was here, one was here, and you have extended in all the directions. Okay, so it could be that this may become a single HSP combining two or more, okay, and this may remain like some gaps are here, and then this could be a separate HSP. Okay, so this is basically the Smith-Waterman algorithm. Okay, right. So this is very important because it is not that each and every seed is matched only one by one. So all the seeds are like thrown on the database, okay, and okay, so that it can be done parallelly, okay, and all the matching and basically extending in both the direction is happening parallelly for all the sequencing in the database. So you can imagine that one sequence has to be searched against like billions of sequences, and you can still get your result in like a few seconds. Okay, so that is only possible when it is done in a massively parallel manner. Okay, so like this was one match that was found, and this was one match, and they are extended in both the directions, and you build on the HSP, right, high scoring pair, and later on if these two HSPs touch, you can continue that as a single HSP. So initially two seeds were there that started the job, and which got extended to both directions, and you build the high scoring pair, or if there were no gaps, like the complete match. Okay, so basically this is about BLAST, how it works. So now how to run it, we will see. So basically there are different versions of BLAST. Protein BLAST. So if you have protein query, okay, amino acid based query, so you will align with the amino acid sequences, that is the protein sequences of the available of the genomes available in the database. If you have nucleotide query, you will align with nucleotide, so BLAST P, BLAST N. BLAST X, like if you have nucleotide query and you want to align with protein database, okay, so we will do the BLAST X. If you have basically a protein query and you want to align against nucleotide, you will do T BLAST N. And similarly, there is a sequence and you want to convert that into 6 reading frames, right, DNA, and then do the translated BLAST in the database, you will do T BLAST X. So different versions are there, depending upon your requirement you can use. Okay, so nucleotide BLAST is one of the standard. Nucleotide and protein are the standard ones, okay, which are used to align a sequence against the database sequences. Okay, so like the page looks like this, and if you just search like BLAST NCBI, you will find this page. Okay, so basically your sequence you can enter here. Okay, it could be a faster format sequence with annotation and the sequence, right. So if you do not have like the sequence and just the accession number of the sequence, because all accession numbers are provided by NCBI, and this is also an application run by NCBI. So if you just write the accession number here, right, so it will retrieve the sequence from NCBI and will give you still the hit. If you have multiple sequences, you can also upload your sequences like this. Choose a file and upload a faster format file. You can give a job title otherwise for your search. Otherwise, if you want to, otherwise it will pick the first header or basically annotation and give the job title like that. Okay, and then you can do 2 sequences alignment or more sequences with this option, right. Then there are some default databases, like generally people do human or mouse, okay, or transcriptome database search and like that. Okay, otherwise the standard one is the NR database search against NR, non-redundant database, that is all the proteins known so far from all the organisms, right. So that you from your drop down box you can choose. Okay, and okay, then basically mostly you will be looking for highly similar sequences, but if you think that your protein could be very different from the available proteins, you can show somewhat similar or dissimilar sequence BLAST also. So that will reduce the scoring criteria, and also change the scoring matrix from like one could be like 6 to BLOSSOM 62 scoring matrix, which is like 62% identical proteins, good for that. BLOSSOM 90 for more than 90% identical proteins, and BLOSUM 30 for less than 30 or 30% identical proteins. So BLOSUM 30 could be used for very low sequence identity options. Okay. Then how many target or hits you want? Of course, the first hit is the best hit, and that is generally taken for annotation or taxonomy. Okay. Then expect threshold E value we will see later. Okay. So basically 10 means that there could be like 10 from all the thousands of hits that you get, there could be just 10 sequences which could appear just by chance. Okay. So if it is 0.001, then basically more you decrease it, like for example standard you can keep it 10 to power minus 6 or 10 to power minus 9, right, or minus 15. So more you reduce it, okay, it is the probability is that is the probability that you can get something by chance. Like 10 to power 9 is the probability, if you keep that as a threshold, that you can get anything by chance. Okay. And the word size default is okay. Generally for these software, default options are always good. Match mismatch score, that okay, how do you want to calculate the match and mismatch, and and the gap cost is linear, so that also you can track down and select. Remove these low complexity regions like repeats, right, or specifically for like different genomes also repeats you can remove. Okay. And this is for example your sequence, right, and you will select a database, okay, accordingly, which I have shown. These are the some of the databases which are available. Okay. So low complexity regions should be removed because they are repetitive, or the probability that they can occur more than once or many times in the genome is very high. So they will obviously confuse your algorithm and the process that we have seen initially, the seeding and matching process. Okay. So they are removed, and basically these repeats are masked or basically converted to n's, okay, in the sequence, and then the remaining sequence is matched with the database. Okay. So the database of choice are generally non-redundant database, which is nucleotide or protein, or basically the SwissProt database. SwissProt will have like more curated proteins, and if you want only against those proteins for which you are sure that this is the annotation and the function, then SwissProt. Otherwise NR is also good for, because it will be huge database, it will take more time also. Okay. Then the expect value is one thing that that we discussed, like how many hits in a database of particular size you can get, you can match your query to by chance. Okay. So generally by default we pick the one which is the highest score and lowest E value. Okay. But how low should be good, like, okay, so basically generally 10 to power minus 6 or minus 3 is also equally good, right. So we will discuss the E value in the next slide also. And the word size is generally default is okay, but if you think that your query and can be easily find, then you can increase this word size. Okay. If you think that it is like very unique and may not be exactly found in the database, then you can decrease the word size. Okay. So expect value is, the definition says that number of hits you can expect to see by chance when searching a database of a particular size. Okay. So for example, if I E value of 1 is assigned, that means I can expect to see out of all the possible hits, like 100 or 200 I have got, one could be I am getting by chance. That means which is not the true hit, but hit that you can get like simply by chance because of the criteria that you have used. Okay. So basically E value of 1 is low. Okay. And mostly, like in all the cases, you will get the expect value in like very low, like less than approaching 0, or basically much less than 1. Okay. So this is only good for like 1 or 10 that by default we are keeping, is only good for the rarest cases when you are not finding any match. Okay. Okay. So this is the output, and the matches that you have found. So you can see the expect value is near to 0, and 100% match you have found with. Okay. So that means your protein that your sequence that you are searching is already present in the database. It has a max score of 2004 and total score of 2017. So basically max score is the score of the best local aligned region, and there could be more than one local aligned region because okay, the match was done partially. So if there are 3 local aligned region, so one could be getting like this score and other 2 are getting the remaining scores like 400 and 217, okay, and this could be like 2004. So this way the total can be 2617. So basically sum of all the local aligned region is the total score, and the best matching region is the max score, and max score is generally considered, okay. And query cover is 100%, that means from head to tail you are able to find that, okay. And there are more options to like view graphically and make a tree of the genomes of your interest. Okay, this we have covered earlier also, how to install Blast, but basically just to give you a quick revision. So basically you have to first install Blast by using sudo, which is super user. Okay, this will ask for a password, so sudo, because you have to log in as root or log in as root. Then apt-get is the way to get it from the internet. Install, right? These are the default command in the Linux, or you can install this package also. And ncbi blast plus, it will search in ncbi and get it, so you can download, okay. Or you can do wget, and this you can get from like the source page of ncbi, okay. Right click, copy, and copy the link, paste it here. So wget and the link, it will get it, uncompress it by using tar -zxvf and this, okay. So it will be unzipping it and untarring it by this -z for unzipping, -x for extraction, v for verbose, and f for files, okay, or folder. And basically then you see that okay, current directory is now Blast, where you have downloaded it, okay. This is the file that you have downloaded. So you should go into that file by using cd into going into that file, then go to the bin folder and then say pwd present working directory, okay. And then you can get a path here, whatever is the path you are getting from pwd, you can do like export path equal to this, okay, to $path, okay. So this way the Blast path becomes your one of your default paths, and you can call Blast from any of the folder in your system, right? You don't have to go to the original path where you have downloaded and installed it, but you can access it from anywhere, okay. So like this is the bin folder that contains so many executables like Blast and BLASTP and so many other executables also, okay. So the executables you will get from this directory, okay, this folder, and this executables folder, and there is a database folder to get the NR and Sysprot and other databases, right, which will be like this in FASTA format. You would like to download like NR and nucleotide or protein or PDB database or Sysprot database, okay. So these are readily available in GZ format. Just download and unzip and start using, okay. And basically this is the executables that I was talking about, RPM, and whereas the complete binaries are also available. Okay, so other way of assigning function to proteins is by eggNOGs is one of the also common way, and it is more based on orthology, because same proteins or similar proteins are present in multiple genomes. So if you know the function in one, you can also derive the function in others. So it's a database that is provided by EMBL, the database as well as the tool, okay. And basically there was a idea of cluster of orthologous groups, like those proteins which are similar in function and also show like orthology that is present in more than one genomes in a decently conserved manner, okay. So those were COGs, and each of those like protein family, that cluster that we made, will be one function, one type of function, and then like similar approach is used for this eggNOGs, which is non-supervised orthologous groups, okay. So basically ab initio trying to make orthologous groups out of these. So this is the database of eggNOG 6.0, and from 12,000 organisms, more than 17 million orthologous groups are available, and trees, and and you can see like right now eggNOG 5 was the previous version, and now eggNOG 6 has come. And there is a tool also, eggNOG mapper, which is very useful, or there are other options to do a few analysis, but like if you do all automated, then this tool is one of the options. Okay, so what it does is, so basically you can paste your sequence in the sequence search box, okay, and it will find the orthology. Otherwise you can use the eggNOG mapper tool, which works like this, that basically you can either give the complete in the next slide, if you can see, like proteins or CDS or genomes or metagenome or seeds in this, okay, and it will first predict genes, okay, and then against the eggNOG database it will do the search, that whatever it has found here in this tab, the genes, it will search in the eggNOG database and then find the orthology, that how many other orthologous organisms or proteins it is matching to. Okay, so that is one part which is good for making phylogenetic tree and also for other information. And for the annotation, it is using its own database of eggNOGs, okay, which can on the COG kind of based annotation database, it will annotate the proteins into various functional categories. Okay, so this is the eggNOG mapper which is available here, and it's an automated way of assigning functions to a protein. You can upload the large files also and select the database of your choice, and it will assign function to the proteins in the specified eggNOG categories. Okay, so this is a paper that covers this and was published about 2-3 years back, right, and October 2021. And basically whenever you use a tool, you should always read the paper that is mentioning that tool, because that will tell how to use it, what is the advancements in that, and the logic and algorithm in that tool. Okay, so what we covered in this lecture is, we looked at what is protein functional annotation and discussed two of the important tools, that is BLAST and eggNOGs, for the annotation of proteins in either genomic, metagenomic, or single genome, multiple genomes, single protein also you can annotate by these methods. And in the next lecture, part of the lecture, we will look at the other methods, that when BLAST does not provide you any hit, then you can use alternate methods for assigning functions to protein, or basically at least knowing that what function it may do in the cell. Okay, so thank you for today. Bye bye.